Let me know when you're ready.
Yeah, that's a good spelling. Which over the slide on this thing, I think.
Okay,
Yeah.
so should we start?
Okay, yeah, so today until Friday, we're really excited to have Yosef Grodzinsky giving a mini course. And he'll be teaching us about neuroscience.
Well, something. Okay. So first of all, my most profound comment probably is that I'm quite deaf. So if you want me to hear you, you're gonna have to scream your lungs out. That's the comment, okay? Hi, Ken Wexler. Good to see you. Okay. So, one question that I ask myself for quite a long time now is who needs experiments? And I think in this department, it's exactly the right place to ask it because it's by and large it doesn't tend to be the most experimental linguistics program in the world, but still, the spirit here respects experiments quite a lot. We have a major experimentalist right in front of us. But of course, one thing to remember, and this is something that I hope has been hammered into everyone's heads for a while, you don't just do experiments. We used to call it the let's do a study kind of approach, like you sit with your friends in a coffee shop and you have this brilliant idea and let's do an experiment. This is not good enough, and I think the one thing I can recommend is the approach of a lazy scientist. The lazy experimentalist doesn't want to do experiments. The lazy experimentalist needs to be convinced that the experiment is going to teach us something important. Otherwise, why do it? And by teaching us something important, at least in my narrow world, what that means is that it would help you distinguish between hypotheses. Exactly in the way you learn in linguistics. And I don't think that what you do is less experimental than what I do. And I think it's an important thing to keep in mind. And once you keep it in mind, it helps you to articulate more clearly what it is that you're really looking in this particular experiment. And what I'll try to do here is to explore the hypothesis that I and about 3.5 others and worldwide have been trying to do for quite a while, which is to say the way we should look at cognition in general is as if there's like small pieces of cognition and small pieces of linguistic knowledge live in small pieces of neural tissue. And the question is, how small are the pieces? Functionally, how small the pieces are anatomically? How do we know what constitutes a piece? And how do we figure out whether there's actually some kind of congruence between them? These are critical questions. And I'd like to start off with several examples, but not before I tell you about what's coming this week. One, I'm going to lay out 4 different approaches to brain-language relations. And I think it's important just because if you happen to stumble upon a paper in neurolinguistics, it might help you map it onto where exactly it belongs, what kind of claim is being made. And the number of claim types is almost as large as the number of authors on these papers. So you better get some feel of what exactly is being claimed and what exactly what the intention is of this particular approach. Then I'm going to give you a crash course in microscopic anatomy, something that I doubt anyone knows here. So it's going to be a little bit more like a teaching mode rather than some dialectic, but more like teaching. And I happen to be affiliated with a very strong group in computational anatomy, and I think it's kind of important to know what these guys are doing in order to teach us what counts as an anatomical module. So who here has heard about Brodmann's areas in the brain? Okay, so several people have heard about this thing. It's very difficult, I think, for any of you to actually articulate what a Brodmann area is. So you know that the brain is parcellated into things, but the question is, how do we parcellate? How do we know that this parcellation has any significance to anything? So I'm going to try to give you a crash course on that. And then when we have the linguistic muscles that all of you guys have, and the anatomical muscle. We're going to try to make these things work together, and we're going to do it in two different domains. One is in a small domain, which is like monotonicity. And my monotonicity lecture is going to be divided into two pieces. Piece number one, which is going to be in this class, and piece number two, which is going to be in the departmental seminar late next week, a week from this coming Friday. And then in lecture number 4, I'm going to talk about a problem that has made many people shy away from neurolinguistics, which is the problem of individual variability and non-replicability and all kinds of bad things that people have been telling you about me and my people of my ilk. And I'm going to try to show you that, in fact, this is based on some twisted up view of what the way the world is. And I'll show you how to look at syntax, very basic stuff like syntactic movement, and pit the variationist approach against the systematic, like linguistic-based approach, and see who wins. And in the last one, which I kind of semi-facetiously called linguistic theory and its enemies, which is a little bit of a takeoff on a book by Karl Popper, The Open Society and Its Enemies. You can see Popper here. I wanted to kind of look at what people like me, inspired by people like you, what we're doing, and pit it against what's happening across the street, basically. Okay, and across the street is one example, but the anti-linguistic approaches to language are quite prevalent, and they seem to be winning in many respects. And I think it's very important to find a way to connect to them. Let's see if we can. So this is the plan. Okay, the 3.5 versions of what it is that one does in neurolinguistics. Version number 1, you just use neurolinguistic data in the same way that you use linguistic data for your theoretical things. You have some kind of a variable, and what you want to do is to see how this variable gets derived, or values, or parameters of something gets derived from a hypothesis. In your case, it usually is some kind of a categorical variable, like grammatical, ungrammatical, plausible, implausible, this meaning, that meaning. In these cases, you use continuous variables like reaction times, like brain waves, all kinds of stuff. And in principle, what you need to do is you use these data to decide between your theories. This is the standard stuff. So, you extract it from some continuous variables. It gives you some novel evidence of the kind that you haven't seen before. The second version is a little bit more neuroscientific, you might argue, in nature, which is, it says, I want the above, but I want a little bit extra, namely, I want some localizing information about where exactly this happens in the brain. And again, there are all kinds of methods. There's the subtraction method, which you can use with aphasia, for instance, and things of that type. There's a piece of the brain is missing. Let's see what's missing from your linguistic ability. But the idea here is you use exactly the same kind of logic as before, except you require of it to have something anatomical in nature. How good this anatomical stuff is a matter of debate, Is it valid? Is it not valid? We'll leave it for later, but the agenda is as I just specified, namely both some linguistic variables and some anatomical location. And the third version is probably the most courageous one. It wants something that goes beyond that, namely it wants linguistic hypotheses to be tested, it wants some anatomical locations, but it also wants to get at the physiology. It wants some neural mechanisms. So people who would do that really believe that you can eventually derive everything there is about linguistic knowledge from something neuronal. It doesn't make you an AI guy, it doesn't make you a connectionist, doesn't make you anything, it just makes you hopeful. And what I'm going to show you are two versions of it, a kind of extreme relative to one another. One is a version from phonology, like an attempt to identify something about phonology, and the other one is a version like to show that you can actually get the big picture straight out of neurons. So we'll see that. And there are going to be 4 papers that I'm going to discuss, at one level or another. One is by Alec Morantz, and I forget the first name, I think Laura Williams.
Yeah.
And from a few years ago on morphology. The second one is an old paper by Angela Friederici and several others on scrambling. A third one is a paper that came out from a fantastic neurosurgery lab in San Francisco, headed by Eddie Chang, about 10 years ago. And the head author is Nima Mesgarani, a fantastic electrical engineer who extracted signals from tissue in an unbelievable way. And finally, this is a paper about deep neural networks by Ariel Goldstein and Uri Hasson and a whole bunch of others at Princeton. That's what we're going to be looking at. So let's just go one by one and see what these things do. Paper, the first one it's a work by Williams and Morantz, as I said, and the question here is, do words decompose in reading, and if so, along what lines? So you can take this question and cut it into 2 pieces. Piece number 1 is, the words decompose in reading, is a standard psycholinguistic question. And the second one, which is more of interest to you, and I think was more of interest to them as well, is along what lines these things get decomposed, and that's a linguistic issue, what exactly are the principles? And there's 2 hypotheses. One is that words decompose by rule, but only when it's transparent. You can just take the pieces and break them, the like superficial morphology kind of approach. And the second one that says words always decompose by rule, and for that, you use MEG, some technique of brain waves that I'm not going to get into because it doesn't really matter. And the important things to dwell on is what is not done here, not so much about what is done, because the logic of this particular endeavor is really the standard stuff that it's like your bread and butter. What's more important is what's not done here. So what is chosen here are particular brain waves at a particular time point during processing, like 170 milliseconds from the onset of the signal. It doesn't really matter. And it's chosen on the basis of past knowledge, which is perfectly legitimate. And the second thing that is also chosen is a particular piece of brain that's called fusiform gyrus, which is what you see over there painted in green. And again, the logic of this experiment is really simple, and you can see it here. It divides the world along 2 dimensions. Dimension number 1 is the rule, so you have like truly complex words as opposed to pseudo-complex words that people are exposed to. And the other one is this dimension of how transparent the morphological decomposition is. It's not the most, it's not the most exciting experiment, but this is not why I chose it to. I chose it to make a point. And there are 2 hypotheses, and I kind of drew the predictions of each. One of them that says it's only in the case of morphologically transparent words that you should get a signal for morphological derivation. This is the green stuff. Whereas the other one that says, whether transparent or abstract, you should always get a brain signal of morphological derivation, which is hypothesis 2. And you can see that you get the left column in this little ANOVA table. This is this little design table. And the result here is that indeed in this green area, as predicted, you could see a sign of there being a morphological complexity carrying over to abstract words like excursion and things of that type. So this is the result. The reason and the conclusion follow that this is an area of the brain that things happen. The reason I brought this is not because of the exciting results here or the exciting design or the depth of the idea. I mean, it's a perfectly fine work, but what I brought the reason I brought it is to say something about its limitations. And limitation number 1 is that there's some kind of a clear but kind of questionable assumption that what you're measuring is directly related to morphological processing. And this is a little bit of a funny assumption. I don't want to get too deeply into it, but just for you to keep in mind that in order for you to actually relate some kind of a brain measure to the morphological, the linguistic computation, you need a mapping. And it's got to be explicitly made. It's not just like any kind of variable that you measure more means something. You need an explicit mapping. The second thing that's important is that the search is very limited to a specific brain area. Now, it's limited in a way that's justifiable on the basis of previous results. But the important thing is to remember that you don't know what happened in other areas of the brain at that given point in time, because you initially restricted your measurement to a specific brain region. So it's kind of weak in the sense that the relationship between what you would like to know about the brain and what you'd like to know about its linguistic activities is limited here. Now, again, this is something that's professed by the authors. It's not as if they're making anything up. No, they're not at all. But this is a clear limitation, and you can see that the goal, in fact, here is just linguistic, which is no small matter. But the goal is just linguistic. There's very little that you can infer from this kind of study to anything that has to do with the neuroscience of language. It's just the use of a different method relative to what you use in your daily life. And the last point is a point about anatomy, which is a little bit of a little pedantic yet important, which is we want to know how this green stuff was drawn. It's not clear at all. It looks very elegant, and when I see elegant things like this, I get suspicious. Because I know people know, my daughter knows a lot about Photoshop. Okay, so I get a little suspicious about such things. And again, I'm not saying it in any critical way of Williams and Morantz, not at all. It's just like important to know these are the limitations of this study. And they would be the first to agree with this. But it's important to know this is just something about linguistics. Question number two, this is like old stuff. It's a study that's about 20 years old, but it kind of makes the point. And the study here is something of the type that used to be done in the 1990s and on in psycholinguistics in German. So there's this thing called, I think, Mittelfeld or something like this, like the middle field, and you can actually cluster, with double objects, you can cluster the subject and both its objects, one next to another, by putting the standard V2 tricks and things like that. And the question here was, would you get an increased signal of the brain's effort as you start playing with syntactic movement? Okay, so this is this middle field, and then you start changing the ordering here by putting one of the objects across the subject, and then by putting both of them. And the question is, would you get some brain index of increased complexity? And critically, if so, where in the brain? This is why I brought it here, because here the question was set up explicitly, not just to find some measure of complexity that would be growing or something like that, but also to localize it in the brain. And lo and behold, it was found to be in Broca's area. So one thing that you can see is like the 1, 2, 3 displacements, or 0, 1, 2 rather, displacements. Yeah, I wrote 0, 1, 2. Okay, I can see it here because this is better. Okay, so the 0, 1, 2, you can ignore the perforated line. It's some average that's meaningless here. And so if you look at it, the question here is, where is the area of the brain in which you find the strongest effect of this 1, 2, 3? It's something called the linear effect, but it doesn't really matter. And the answer is yes, in Broca's area. Great. So what have we done? We've found some kind of a brain correlate for some syntactic process, and we localized it in a single place. The important thing here is just that, namely you localize it in a single place, because here, unlike the previous case, you did not pre-select a specific piece of the brain And this pre-selection is usually done for all kinds of statistical reasons having to do with multiple comparisons, and it's basically signal to noise issues. Here, there's no pre-selection. You do a whole brain analysis and you find something, and you can, at whatever reliability that those measurement instruments allow you, you can actually say, this is it, this is the place. The place where the activation is most important, significant, if you will, okay? But still, you know, you can say all kinds of critical, you can, what's happening here? Oh, okay, right. So now there's several questions and points to note. First of all, we know nothing causal here. That's obvious. Okay, we know that, yeah, we found something about syntactic movement in this area, but what exactly is the relationship between the brain signal and remember, the brain signal is a fairly remote index of neural activity. If you want, you can ask me later about it, but it's a fairly remote index. And the second point to be made, which is this is like a considerable agreement between this result and aphasia results in the past, namely the same area that is more strongly activated is an area that once lesioned loses the ability to properly process displacement, like syntactic displacement. That's like the roughest way of putting it. So there's some kind of a nice convergence, if you would. It's not bad. And what you really have to see, and this is just a small critique of this particular paper, there are behavioral measurements like error rates of the subjects and reaction times. And when you start looking at them, you see that they don't really, the relations between the different conditions do not mirror the relations that you have on the imaging domain. So if you look at the signal domain and the way the signal gets organized by complexity, and then you look at the behavioral domain and you look at the way the reaction times get organized by complexity, they don't match in this particular paper. Okay. I'm mentioning just for one reason, that every time if you ever get the good fortune of reading such papers, you really have to be careful and make sure that if there's several measures, they're consistent. And often you will find that they're not. It's just human. And finally, the question is, I put Broca's area in quotes, and there's a reason for it, because when somebody tells you about Broca's area, and I'm sure that more people in class have heard about Broca's area than those who have heard about Brodmann's areas, okay, you have to ask them, what exactly do you mean? It's a good trick, because even if you don't know what it means, you can almost be sure that your interlocutor doesn't know either. And it's really a good trick because it makes people reflect for a moment of what it is that they're talking about with respect to brain areas. Okay, we're now moving to I think the most exciting piece here, which is the Mesgarani paper, which I think is I personally think it's an incredible paper. And so, and the question here is, given that we know that the superior temporal gyrus, which is, I don't think this is working for me. So this is the superior temporal gyrus, which is very close to auditory cortex. In fact, it overlaps with it. This would be auditory cortex. Would this superior temporal gyrus know anything about speech? And what exactly would it encode when it comes to speech? And what these people do, technically, is Abby Chang is a neurosurgeon in San Francisco, and he operates on people who have epileptic foci that cannot be controlled by medication. And the way you do it, I'm sure that you read about it in many places, you open the skull, you put a bunch of electrodes, a matrix of electrodes, small electrodes on cortex, and for about 10 days, you test your subject on all kinds of behavioral tests, and you get a mapping of what exactly is the function of each little part there. And this helps you to navigate during surgery. So you actually use the subjects as a navigation device. I didn't mention before that if we're gonna have a little time, I'll tell you what we do. I work in Düsseldorf at the medical school, and I help people who do a malignant tumor resection with cancer patients. And we use the patients during the operation. I actually go to the operating room and I test the patient as they're in awake surgery, as they're being operated on. We use the patients as a navigation device. You put it's an old method that has had like a second life, but you use the patients as a navigation device. And if you see that the particular area is overly active, you stay away from it, especially when you have cancer patients whose life expectancy is not very high. You want to make sure that, whatever is left for them to live, they're going to have quality of life, which is the top principle here. If you take their language, it's not good news. So, when the contribution of people like me is to give better tests with linguistic tests that have better resolution than what's standardly used. Here, it's the same kind of trick, except you do it pre-surgically because there's, you can put a bunch of electrodes beforehand, and then you can actually see which function or functions you can attribute to each little brain area. And on the fly, you can do all kinds of experiments. So what they did was they asked Keith Johnson, a phonetician at UC Berkeley, they asked him to give them a list of words that is full coverage of the whole phonetic inventory of English, which he did. And they gave several voices, male, female, and this was a way to try and scope the broadest spectrum of speech signals that you can imagine. And then Nima Mesgarani, as I said, an incredible electrical engineer, just took it and did something with it and managed to extract signals from it, which I think is like sci-fi, honestly. And so what they ended up doing, and this is an example of just 5 electrodes, they extracted with all kinds of noise reduction techniques, they managed to extract signal from specific electrodes. You can see 5 of them here. And what's interesting about the signal intensity is that it clusters by distinctive phonological distinctive features. This is the miracle here. Okay, it structures by abstract features. And they then went on and they did hierarchical clustering. You can see it here, how these things cluster really by features. Here you can see how it goes by sonorant and obstruent, and it basically clustered fantastically by abstract phonological features. I think it's a miracle, In fact, a friend of mine and I wrote at the time, we were asked to write a commentary to Science, and I said, this is like you take Roman Jakobson and Nikolai Trubetskoy from the 1930s, and you read it straight out of Cortex. This is what we said, you can take a look. You read an old phonological theory out of Cortex. It's what better miracle can you imagine? Yes.
Can you contrast this to the last paper where you said we can't?
I can put.
No, I'll just, is this better if I go like that? So the last paper you said had no causal, you couldn't do causality with it.
Right.
This, it seems like we're concluding something causal from it. And I think this is a sort of slippery trick that happens across neurolinguistics, where in one type of study, you have correlative stuff that is obviously non-causal. And then in another type, there's correlative stuff that seems to be linguistically causal. Yeah, right. So okay.
So you agree with number 1. So we agree. I didn't think anyone claimed that there's a mechanism here, but it gets closer.
What do we mean by implement then?
You just know which areas, look, descriptively speaking, no, these are great questions, but descriptively speaking, that these areas are most active during the processing of this particular feature. And the level of their activity is pretty much identical across all instantiations of this feature in the specific consonants at issue. This is the only thing that you know. This is what the data allow you to conclude. The question is, what exactly does that mean? So suppose, let's just take it one step further. Suppose I told you that I know about the anatomy of this. Suppose I told you, which is against what's happening here, parenthetically, suppose I told you that each distinctive features has its little anatomical corner. So, you know, you have 10 electrodes that share this kind of behavior, and they're all adjacent. And they form a little cluster. You know, it's not a mechanism and it's not a causal explanation, that's for sure, but it brings you a little closer. And the sense at which it brings you a little closer is because you have better understanding of the nature of the signal, because this is the physiology, it's not some remote index like functional MRI. And the anatomical resolution is way better, you just put it there. So the number of neurons that are involved is much smaller than elsewhere. In this respect, it just gives you a clear picture, but it's the blunt, I do agree with you completely, that it's still the blunt localization, the mindless localization, if you will, as before. I agree with you, but the resolution is bringing us somewhere. This is the hopeful point.
Okay, so no causality, just higher resolution. Okay.
Yeah, but boy, is it ever. Okay, now, having said that, let me tell you that one critique that I have of this paper is that we know nothing about the anatomy. Namely, what I did tell you was wishful thinking. They do not report where the specific electrodes were placed relative to one another. I think they had about 100 electrodes, and they don't report it. And I'm sure that they would have reported had they known about anatomical clustering of electrodes. They don't report it, and I think because they just, you know, the electrodes were here and there. So this is the downside, but this is where we are.
Can I ask a question?
Yes, Runti.
Yes. So I think in this paper, they interpret the result by distinctive features and natural classes, which are mainly based on the place and manner of articulation. It is a little surprising for me to see this result in the auditory cortex. So I wonder, is this the only way to interpret the result? Can we do that to interpret the result by acoustic cues like F1, F2, and VOT and this kind of things?
So what you're asking, you know, again, with my hearing, what you're asking is basically whether there's additional data from some other aspects, and the answer is I don't know. It's hard enough to do this kind of stuff, so it's not as if they could, to my knowledge, I don't know that they did anything beyond that, but I may be wrong. I don't follow this extremely closely, and this paper is 10 years old. So
Yeah.
the answer is I don't know.
Okay, thank you.
Okay.
Ken Wexler also
Could
has-
I ask a question? Hi, can you hear me? Hi. A fascinating result, you know, and of course it's, in a way, it's like a linguistic equivalent of Yubu and Weasel, visual. But one thing is, has it been followed up, which might be an interesting way, as I understand it from the very little bit of phonetics that I know, that I think I recall that the exact parameters of how voicing is done acoustically are different in different kinds of language. That voice onset times are different, not exactly the same from language to language, and even some other factors, I forget exactly. And something that would really be beautiful to see, has it ever been tested since then? How it might look in some other language where the acoustic parameters are different than in whatever language this was done in, I assume English, but.
So my answer is to you is in number 3. Namely, the activation maps are not entirely modular. They interact with auditory factors. And one factor that they do interact with is voice onset time. So there is I don't think that they pursued it any further, but there's clear interactions. It just makes it makes the issue, it makes the issue more difficult. Maybe I should shut my, I don't know how to do this, never mind, because I keep seeing myself. But,
Stop it.
no, yeah, but. Never mind, listen, I'm such a zero when it comes to technology that I don't want to embarrass myself even further. So Ken, the answer is yes, it just complicates matters. We're focusing here only on that, mainly only on the fact that abstract distinctive features as opposed to anything concrete, in fact, has direct representation. That's the only thing we're focusing on. The fact that it interacts with other factors complicates matters. So let's keep them aside. Okay, last question, and then we should continue.
Yeah, just so I'm clear, this paper is, I guess you have it because it's representative of the third,
Yes,
the third way of doing neurolinguistics.
exactly.
I.e. that there was some mechanism underlying.
Well, but it's closer.
Closer, do we have examples of, I mean, maybe the next thing you're gonna show is the mechanism bit, but like, do we have any sort of, mechanistic understanding of,
Well, this is the next one, at least that's the claim.
okay.
Thank you for leading on to the next one. Yeah,
quickly.
Say
So
again.
I think this group have some papers on tones, and that one I think reveals more about the mechanism.
I think everybody's worried about mechanisms here. There's no question. I would be the first one to worry. But the thing is, the question is what it is that we can do. So I suggest we just continue, okay? All right, so here is number 4, and this is, I don't know your name.
John.
John's, we're getting into the real thing, okay? And this is a paper from a couple of years ago by Ariel Goldstein, who happens to be my colleague now in Jerusalem, and a whole bunch of others at Google Research in New York and at Princeton University. And the thing is, what are the brain correlates of sentence comprehension? This is as big a causal picture as you want. Okay,
Correlative.
maybe not big enough. I don't know. But this is like, this is the idea of a mechanism. Okay, and the claim here, and it's important to see, is that there's a task that's called next word prediction, which is the best index there is for anything we know about language. And what we need to do is to model it. So here's a research program. We give you some language. We take measurements of the best performance regarding next word prediction from some kind of GPT. That's number 1. We get the best measurements of performance of humans, namely we give them a task to predict the next word. And we get the best measures of functional MRI of some brain region or some brain method of some sort. And if we find high correlations between the brain's abilities as measured by whatever electrodes and whatever way you poke the brain, and behavior, and then the third guy, which is the machine, this is your mechanism. GPT-2.
But it's correlative.
Sure, but this is as close as close, you just derive the behavior.
Okay.
The logic would be, if I were Ariel Goldstein, I would tell you, look, I'm giving you a machine that has a particular behavior. It mimics humans in this particular, on these measures. It also mimics the brain. What else can you ask? Nothing. This is as good as it gets. I'm not being facetious. I'm trying to represent these guys as judiciously as I can. But again, this is what the market avails us of. And as you expect, step number 1 is to take some text, divide it into small windows of 10 words, and every time you move this window, you ask your subjects to predict the next word. And then you look for, what word it gives you the highest percentage or highest proportion of predictions. And this is step number one. Step number two is you do the same thing. Step number two is you do the same thing with GPT-2. You give it a window and you ask it to predict the next word, and then it gives you the top prediction, and then you just set up a correlation. So you get some quantity of how, what percentage or what proportion of times did it predict milk, and then you compare it to what percentage of time did your 500 humans predict milk, and then you set up a correlation line, and this is what you get. And then this is step number two. The text, I should mention for those interested, is some kind of a very noisy file from a story in This American Life about a gorilla that grabbed somebody's camera and started taking pictures somewhere in Africa. It's extremely noisy. It comes with the New Zealand accent of the photographer and American accent of the entertainers, like This American Life, and a lot of jungle noises in the background and what have you. And then this is what you get. And of course, the behavior of humans, if you want to be really critical, it comes out of written experiments, all kinds of issues, okay, but it doesn't really matter. This is the logic. This is it. The correlations are high, it's beautiful, and the next thing to do is to go and have the brain listen to this. And again, they use the same kind of data that was used previously, namely neurosurgical data that were collected by neurosurgeons and by some 1,300 electrodes on somebody's brain, 9 patients, I think, altogether. So the 1,300, I think, are the collective of all 9 patients or something like this. And they found that 160 of these belonged to the class that had high correlations both with brain and with machines. This is it. And this actually comes with a claim that you've been waiting for. And the claim you've been waiting for is that the human brain processes incoming speech similarly to an autoregressive deep learning. This is it. And I'm just a messenger. Okay.
But do you think that's causal? Like, I'm sure they do.
You know what? Oh, if I think this is causal,
Yeah, I'm sure nature did, but.
I don't want to put myself in a position to defend this thing or even attack it. I'm just saying this is the claim. And if you ask them, is it causal? They would say, sure, this is the best model we have. And I measured it. They would even tell you against 47 different machine learning models. This is autoregressive. The other one is monoregressive. I don't know. Okay, this is what the market contains. And, if you want to have some critiques is of course, there's this idea that in fact, Uri Hasson, the leader of this group, really deeply believes in my view, totally mistaken view of what science is that science needs to be done in the wild. There's this thing called in the wild, like, you have to run around with a microphone and collect utterances in class or something like this. This is the only way. He truly believes in it. In his justification, he got, I think, the wrong education. A friend of mine a very good friend of mine was his thesis supervisor, Raphael Malak at the Weizmann Institute in Jerusalem, in Israel, a fantastic scientist, but somehow this is what happened. And he really believes, he truly believes that this is how you should do science. Just collect sentences in the wild, and NPR is a good wild, so you go to NPR or something like that. So this is point number one. Point number two is there's no anatomical information at all. In fact, about 2 months ago, Ariel Goldstein, the first author, presented it, and I asked him, where did they get, because they make claims about Broca's area. And I asked him the standard question, because I'm a boring guy, how do you know it's Broca's area? And they said, oh, the neurosurgeon said it. And it turned out that the way that they did something that I personally never do. They got the data files from the neurosurgeons. They never saw a single patient. They got the data files, and there was one column where the surgeon wrote his or her impressions in surgery of what brain area this was. Now, it's quite difficult. I don't know if any of you has seen brains separated from their owners and then seen brains inside their owners. It's very different. I don't know, you know, I spend time in operating rooms and I can hardly, I think by now I can identify motor cortex, which is very easy. Okay, it increases my admiration to neurosurgeons, but it's very, very difficult. And the level of certainty that you get from this column is very low. So what do we have here? Nothing much. Okay, but this is as close as it gets. So this, if you look at it and you organize it, and I think it's really an important map for you. Every time you read such a paper, you just say to yourself, where am I? What claim is being tested? In the Williams paper, Williams-Morantz, what's being tested is a linguistic hypothesis. That's perfectly legit. That's the right thing to do. But this is as far as it goes. In the next one, you know, again, forget about the quality of data, the precision of the question, whether focus is or is not involved. Forget about all these things. Just look at the logic of inquiry. This is something about localizing claims. And for that, you need at least two additional things, you know, other than what was discussed. One, you need a clear concept of brain loci, a clear concept of that, and what constitutes a brain locus. And secondly, you need to make sure that you examine the whole brain. These are things which are important. And then perhaps the strongest, John would object, but I think it's still stronger just because it gives you much better resolution, is the Masgarani paper that showed this incredible thing of, like, even at the physiological level, when you stick in electrodes, you still find highly abstract features. And with respect to the Goldstein paper and this GPT stuff, I don't think it's particularly helpful. So this is chapter 1. Let me take a few minutes and jump straight into chapter 2, because I think we're a little bit squeezed for time. I'm not going to go, you know, more than 3 seconds beyond my time. I don't like it because people hate you when you do this. But you know, what I want to do is, again, pursue this hypothesis, namely small anatomical pieces of the linguistic homunculus. So you can think, you know, all of you heard about the homunculus, this representation of sensory motor cortex, of sensory motor abilities in cortex, where each organ is represented disproportionately. So the claim goes in different places. You can think about different homunculi like this, and people have been mapping all kinds of abilities in different brain areas. And I would like to think about the linguistic homunculus, namely trying to take and parcellate all our linguistic ability into small pieces. And the only thing that makes this special is, one, the resolution, the hope for serious resolution. Two, the insistence on both anatomical and linguistic and linguistic precision. And three, and I think it's important, the insistence of my personal pursuit of knowledge. It's not like processes. This is, you know, what I'd like to do is to put a finger on linguistic knowledge. You know, some people might tell you, might think it's crazy, but I think it's important. And if it would resonate anywhere on this planet, if it should resonate well, it should be in this group. I don't know if it does, but at least it should. Okay, so what do? We go and we try to identify the right natural class in anatomy. And to my mind, it's something that's called cytoarchitecture, the way cortex looks microscopically, and we're getting there in just 3 minutes, to identify the right natural classes in psycho or in linguistics, and then to try to find congruency. And there's a bunch of methods: histological structure, linguistics, deficient linguistic knowledge, and functional MRI, just because it's the method that I've been using, not because it's particularly important. And all of you have seen the brain and know that it has like 2 hemispheres, and you can look at it from different angles. And if there's one thing that should impress you when you look at this, is that even at this level of very graphical stuff, you can see that the dream of hemispheric symmetry is not exactly right. The two hemispheres, and they occupy largely the same volume, but they don't look the same. They look roughly the same, but not quite the same. You know, look here. This is not exactly my idea of sameness. You know, no, it's like important to realize there's a lot of variability, and this variability causes huge measurement problems and mapping problems. So this is why I'm bringing this thing. And you all know that the brain has, well, 4, maybe 5 lobes. So there's a frontal lobe, there's a temporal lobe, there's a parietal lobe, there's an occipital lobe, and some people actually would tell you that there's something called the 5th lobe. You can see here it says insula projected to surface. There's a piece of cortex that when you slice, you'll see it in a few minutes. Once you slice, you get slices of the brain, seems as if it's isolated, it's not connected elsewhere. Hence, insula, it is called. And some people call it the 5th lobe. I'm emphasizing it because some of the work that I'll be talking to you about actually comes from the insula. So these are the lobes of the brain, and you know, you can identify organs by dissection. So dissection is something that is non-planar. You just dissect something. So here, for instance, the dissection is of the corpus callosum, the big bundle of fibers that connect both hemispheres. You cannot dissect it and stay planar because, you know, it's not as if the brain abides by the internal division into organs, into brain organs, abides by some geometric rules. It doesn't, of course. So here we can see little organs, and the dissection must be macroscopic. In fact, if you could only see the tools that these guys use, you'd be shocked. Okay, they don't even use knives. You know, they use these very, what's the dull, very dull instruments to scrape off cortex, to map, to really expose structures. It's very interesting, in fact. So these are little small organs, and the organs that you probably all heard about is Broca's area and Wernicke's area, and you know, the premotor cortex, perhaps, things like that. And I want you to note that this is like pure Photoshop, nothing else. And it's really important to realize that all these pictures that you see don't necessarily represent anything. And one thing that you see is you look at 2 brains. So this is a brain from a classical paper, which is almost 50 years old, by Norman Geschwind, the person who revived, almost single-handedly revived the study of brain and language in the late 1950s, after it had been dead for almost a century. And did great things with it. And this is the paper, this is taken out of a Scientific American paper by him, and this is Broca's area. And this is a work that's done by anatomists at McGill, an ex-colleague of mine named Michael Petrides, to identify pieces of Broca's area. And one thing that you can see is this. First of all, the 2 brains look different. One is a little bit flatter, the other one is more spherical in character. Secondly, why should those macroanatomical landmarks like gyri and sulci, which is what is typically used in order to identify organs, why should they be relevant? You know, it's just we know that, you know, during embryonic stages, the brain starts folding. So why should folding patterns have any functional relevance? Maybe it's just a consequence of their squeezing into an enclosed space called the skull. As you just, you know, squeeze more material, it gets squeezed, and because it gets squeezed over millions of years, it gets squeezed roughly along the same lines. But why should it have any functional relevance? A third important question is, even if we take this topographical science, how do we define the location of the border? And one thing that you will see soon is that between 1/2 and 2/3 of cortex is in fact buried inside these crevasses, these sulci. So where do we draw the line? Is it on the right hand of the gyrus or the left hand of the gyrus? Where do we do this and why? We need some principles in order to parcellate pieces of cortex if we want them, and we need to formulate them in a manner that would also make them functionally relevant. Otherwise, why bother? You know? And yeah, and lastly, are there borders which are not defined by those sulci? And here you can see examples of borders which are set up in a way that does not respect those sulcal boundaries. And I think with that, we should probably, everybody has to go. This is the worst thing, is like people start walking out on you. So we should continue with this tomorrow. Thank you, bye.